Aloha everybody. Last time I ended with a brief introduction of the beta oxidation pathway. To repeat that, this is a pathway that degrades long hydrocarbon chains, usually supplied from fatty acids, by removing 2 carbons at a time in a sequential manner, starting from the carboxyl group of the fatty acid molecule. These 2 carbon compounds are in the form of acetyl-CoA. All the resulting acetyl-CoAs are then fed into the citric acid cycle, where they are degraded to carbon dioxide and electrons. The other product of beta oxidation are electrons loaded on electron carriers. All electron carriers from all degradation pathways are then fed into the oxidative phosphorylation part of the cellular respiration, where they're used to synthesize ATP and to convert oxygen with the help of the protons and the carried electrons to water. Alright, so a fatty acid is degraded 2 carbons at a time in a sequential manner, starting from the carboxyl group to acetyl-CoA and electrons. So here's the question then. How many times beta oxidation needs to repeat in order for the stearic acid to be degraded completely? Think about it. Now, in order to answer this question correctly, you need to remember from 481 how long is the stearic acid. And if you remember correctly, or if you check it out, you will find out that it's 18 carbons. Alright, you can do this basically 2 ways. Either you can write down 18 carbons and start removing 2 carbons at a time and figure out how many acetyl-CoAs you're going to generate and how many cycles the molecule needs to go through to be completely degraded to acetyl-CoA, or you can do it in your mind. And if you do it correctly, you will realize that you need 8 cycles, so 8 repeats of the beta oxidation cycle. And why is it not 9? Because in the last cycle, where you have only 4 carbons in the hydrocarbon chain, the removal of the 2 front runners, in a way, the alpha and the beta carbon, will also result in an acetyl-CoA formation of what's left at the end. So in the last cycle, you actually generate 2 acetyl-CoAs instead of 1, which is the product of every other cycle. So the way to calculate this easily is number of carbons in the fatty acids chain divided by 2 minus 1. So 18 carbons divided by 2 is 9, minus 1, 8. So that's how many cycles are required to degrade a fatty acid. If it's palmitic acid, which is 16, which is the most common fatty acid in the human body, so 16 divided by 2 is 8, minus 1, 7. So it takes 7 cycles to fully degrade palmitic acid to acetyl-CoA and electrons. Alright, so now let's look at the beta oxidation pathway and the steps it involves. Now, before we get into the names and the enzymes and the metabolites, I just want you to simply look at the pathway and figure out what happens to the fatty acyl-CoA molecule in order for the acetyl-CoA, for an acetyl-CoA to be released and the rest of the molecule to be ready for another round of oxidation. So what does really happen? Okay, so first the molecule is oxidized between the alpha and the beta carbons. You can see how, and the alpha carbon is the first carbon after the carboxyl carbon, so between these two. And even though it's called beta oxidation, at first the oxidation doesn't appear to be only to the beta carbon, because as you can see, the double bond is shared between the alpha and the beta carbon. What is the oxidative state of these carbons compared to these carbons? Well, each one of them is being decreased by 1. Alright, so we first oxidize the molecule. Where are the electrons loaded? On an FAD carrier, which is part of the acyl-CoA dehydrogenase enzyme. This carrier is part of acyl-CoA dehydrogenase. Okay, it's a prosthetic group. The next step, what do? We add water. We basically added oxygen to the beta carbon, hence beta oxidation. Now the oxidative state of the alpha carbon was restored just like at the beginning, but the oxidating state of the beta carbon is now decreased by 2 electrons if you compare it to the beta carbon in the beginning. We just added oxygen to it. Then what happens in the next step? We oxidized this compound, the beta hydroxyacyl-CoA, and we took another pair of electrons from this carbon, leaving behind a carbonyl group. So what's the difference in the oxidative state of this carbon compared to the initial? 2 electron pairs, or 4 electrons total. But the resulting carbonyl group will enable now the CoA to come in with the help of thiolase, bind the carbonyl carbon, and release. It will bind right here, and it will release the acetyl-CoA, which is right at the beginning of this molecule. So if you compare the first acyl-CoA with the last acyl-CoA, the difference will be 2 carbons, that the last one is 2 carbons shorter compared to the first. And what happened? We have dehydrogenation, or oxidation reaction, loaded electrons on FAD, then hydration, then another oxidation, but this time the electrons are loaded on NAD plus for NADH. Then we have the thiolase enzyme add a CoA, thus breaking the carbon-carbon bond, and acetyl-CoA is released. Now this molecule is ready to cycle back for another 2 carbons to be released in the form of CoA. Alright. Now all these names actually make a lot of sense. This trans delta 2 NOL-CoA, that describes the double bond between the second and the third carbon of the chain. So that's why, and the orientation of the double bond is trans, so trans delta 2 NOL-CoA. Then NOL-CoA hydratase, water is added. And we have beta hydroxyacyl-CoA, hydroxyl group on the beta position, beta hydroxyacyl-CoA. Okay, this is another acyl-CoA. Then we have beta hydroxyacyl-CoA dehydrogenase to form beta ketoacyl-CoA. So instead of a hydroxyl group at the beta position, now we have a keto group or carbonyl group at the beta position. So beta ketoacyl-CoA. Then thiolase adds CoA, and we end up again with acyl-CoA, and acetyl-CoA is being released. So all these names make a lot of sense. If you just spend the time to remember, just to make sense of them, it's not going to be that difficult for you to remember them. Alright, so how many electrons are removed in the process? What is their carrier? And what is the ATP equivalent? 1 electron pair, or 2 electrons. 1 electron pair here on FADH. 1 electron pair loaded on NAD+. What is the ATP equivalent? For every FADH2 carrying an electron pair, the equivalent is 1.5 ATPs. For every electron pair carried by NADH in the mitochondria, it's 2.5 ATPs. So 4 electrons, 4 ATPs. You can remember it either as 2 electron pairs and 4 ATPs, or you can remember it as 4 electrons, 4 ATPs. Okay. Per cycle. So for the complete degradation of the stearic acid, how many total electrons or ATP molecules will be generated during the beta oxidation of the entire molecule. So if you remember, stearic acid requires 8 repeats, each generating 4 electrons or 4 ATP molecules. So 8 times 4, 32 electrons, 32 ATPs. Okay, how many total ATP molecules will be generated for the complete degradation of the stearic acid to carbon dioxide through beta oxidation and then the acetyl-CoA through the TCA cycle to carbon dioxide? So let me repeat the question again. I think I might confuse you a little bit. So how many total ATP molecules? Oh, sorry. I think I just went ahead. So how many total ATP molecules will be generated for the complete degradation of the stearic acid to carbon dioxide through the TCA cycle? And just to clarify, no carbon dioxide was released during beta oxidation. So the only way to break down of stearic acid to carbon dioxide is through the TCA cycle. Alright, to answer this question we need to figure out how many acetyl-CoAs would stearic acid generate through the beta oxidation pathway. 8 cycles, the last cycle generates 2, so we have 18 carbons divided by 2, 9 acetyl-CoAs. Alright, so we will have 9 acetyl-CoAs. The question would be, how many ATP molecules are generated per each, the equivalent of how many ATP molecules are generated per each TCA cycle. Well, if you remember from last chapter, each cycle of each TCA cycle generates 3 NADH, 1 FADH2, and 1 molecule of ATP. So 3 molecules of NADH, that's equivalent to 7.5 ATP molecules, because each electron pair carried by NADH will lead to the generation of 2.5 molecules of ATP. So 3 times 2.5, 7.5. Each electron pair carried by FADH2 would lead to the generation of 1.5 molecules of ATP. So 7.5 plus 1.5, 9, plus 1 molecule of ATP generated during the cycle, total 10. So 10 ATPs are generated per cycle of the citric acid cycle. So we have 9 acetyl-CoAs, so 9 TCA cycles, total of 90 ATP molecules. And then what is the grand total ATP count for 1 molecule of stearic acid from the moment it enters beta oxidation to the time when it's fully degraded by the TCA cycle to carbon dioxide? 32 plus 90, 122 ATP molecules. Now, if we subtract the ATP that we used for the activation of the fatty acid to acyl-CoA and the breaking of the pyrophosphate, which is basically what is here, right here. We spend a molecule of ATP right here, and then the pyrophosphate breaking down. This is the first step of the Carnegiean shuttle. When we subtract that, we'll end up basically with 120 ATP, or high energy bonds, are generated for the complete degradation of 1 molecule of stearic acid to carbon dioxide. Alright, I hope you were able to follow all that. The example in the book is about palmitic acid, so I didn't want to give you exactly the same example, so I just wanted to stimulate you a little bit by giving you a slightly different fatty acid. And since probably most of you like beef anyway, and beef is from the name stearic acid, which is the most common fatty acid, I guess, in beef. I shouldn't say most common, but it's a very common acid in beef. Just to figure that out for yourself. Okay, similarly to glycolysis, what we did then, the maximum energy of oxidation of palmitate is 9,800 kilojoules per mole. If we use the standard versus the actual energy change for the complete degradation of palmitate and the synthesis of ATP, you will realize that based on the actual free energy change, about 57% of the palmitate oxidation was saved as ATP. How much was for glycolysis? About 60. So you can see, whether we degrade fat or whether we degrade glucose, about 60%, roughly, give or take a few percent, are invested into ATP synthesis. The rest are released as heat. Alright, so we talked about the even carbon number fatty acids. And more specifically, we talked about saturated fatty acids. However, in our diet, we also have a lot of unsaturated fatty acids. How the degradation goes for them. Is it different? Or is it the same? And if it's different, how different it is? Let's take a look. Alright. Before we actually get into the details of the degradation of the unsaturated fatty acids, which could be mono or poly, I'm just showing examples here. I wanted you to think about this situation. And this really works well in class, but I hope you will appreciate that, even though we're doing it online. Well, the first step in beta oxidation of saturated fatty acids results in the formation of an unsaturated fatty acyl-CoA right here. You can see it. Trans delta 2 NOL-CoA. It is a unsaturated fatty acid. That's the first step in beta oxidation. So how does that fit with the degradation of common unsaturated fatty acids? Does that make it easier, or does it make it more complicated? What do you think? Well, the first thing you need to realize is when you look at this structure, for example, and this structure is, this is a cis orientation. And it's between, starting from here, always starts at an odd carbon towards an even carbon. This, on the other hand, is a trans double bond, and it starts at an even carbon towards an odd carbon. So, for example, this is delta 9. And notice all these deltas are odd number carbons. 5, well, this is an even, I guess, but it's just one exception. 5, I guess there are some that are even numbers, but most of them are 9, especially the most common one, oleic acids. And this is an even number, and it's trans. All these are cis, whether it's an odd or even carbon, they're all cis. So there is a problem, and the first step of beta oxidation doesn't make it easier or doesn't make it better fitting for the unsaturated fatty acid to fit into the cycle. So for every monounsaturated, we need one additional enzyme to be able for the cell to be able to handle it. And for the polyunsaturated, we need 3 additional enzymes and a molecule of NADPH. So the degradation of the unsaturated fatty acids is actually more complicated compared to the saturated fatty acids. So let's take a look now. Okay, so this is the degradation of oleic acid, or oleoyl-CoA, basically activated oleic acid. And of course, everything will go well, just like normal beta oxidation will go, for 3 cycles until we get to this point. And when we reach this point, then this now compound does not fit in the active site. Instead of having, normally, beta oxidation would lead to this bond, the trans delta 2 NOL-CoA, but instead we have trans delta 3. So it's not in the right it's not in the right position, and it's not in the right orientation. It's cis, the other one is trans. This is cis delta T instead of trans delta T, delta 2. So we need an isomerase that will convert the trans delta 3, the cis delta 3 to trans delta 2, or delta 3, delta 2, N-acyl-CoA isomerase. The product now will be trans delta 2. It fits right into the active site, and then we'll continue with the hydration, etc., etc. Now, the situation with polyunsaturated fatty acids is slightly more confusing. Well, complicated. I shouldn't say confusing, but complicated. Beta oxidation goes on while we have saturated hydrocarbon chain. When we get to the first the first double bond, I should say, which is again in cis orientation, we need the same enzyme, delta 3 to delta 2 N-acyl-CoA isomerase. This double bond now is shifted and converted into trans delta 2. Then beta oxidation continues until we reach the second double bond, and then we end up with this structure where we have a trans delta 2 that is followed by a cis delta 4. Now, a trans delta 2, it's okay. This is actually in the right, the double bond is in the right orientation and everything can go well, but this compound does not fit in the active site because of this other double bond in the close proximity. So we cannot really continue, the enzyme cannot really continue with breaking of this bond. The way the body solves this is by reducing these two double bonds, in which is one cis, the other one trans, into one trans. So we have 2,4-dianoyl-CoA reductase, and we use one molecule of NADPH here. We take an electron pair, donate it, invest it into the compound to generate one trans delta 3 double bond. It is trans, which is good, but it's in the wrong position. Instead of being delta 2, it's a delta 3. So we're going to need to have an isomerase that will move the delta 3 to delta 2 position. It's not the same enzyme though, because this removes a cis double bond to a trans double bond. This has to remove a trans double bond in the third position to a trans double bond into the second position. Alright, when we get to this point, now that fits right into the active site of the hydratase enzyme that will continue down to the next step of beta oxidation pathway and eventually release the rest of the acetyl-CoAs. So you can see that we need one extra enzyme and three, this one, and this one, three additional enzymes to handle polyunsaturated fatty acids and an electron pair from NADPH. So, which fatty acids give more energy, saturated or unsaturated? Now, you may see this more obvious here, and you'll say, well, we invested an electron pair here, so yeah, that's going to steal away an electron pair from the oxidative phosphorylation, and you'll be right, that's true. But there's something else also that you may or may not realize, is that this is, if you think here, the first product of saturated fatty acids is to remove an electron pair from between the alpha and the beta carbons and load it on FADH2, on FAD for FADH2. That will not happen with the unsaturated fatty acids. This double bond will be isomerized, but that step of the first dehydrogenation, the first oxidation step, will not happen for unsaturated fatty acids. So per every double bond, we're going to skip that first oxidation step. So the more double bonds are in a molecule, the more electron pairs will be basically not produced. And not only that, but for every, other than for the first one, for every additional double bond, we have to invest another electron pair. into the polyunsaturated fatty acids. So the easy answer would be that saturated fatty acids carry more energy compared to monounsaturated fatty acids, which carry more energy compared to the polyunsaturated fatty acids. Again, why is that? Because saturated fatty acids carry carbons that are fully reduced in their most reduced form found in the polymer. Again, it's all about electrons. Which compound carries more electrons? The more electrons the compound carries that can donate these electrons, the more energy we'll be able to generate. All right, now we'll transition to odd number fatty acids. Everything goes as described before. If they are saturated or unsaturated, they'll be handled until we reach to this last product, which has 3 carbons, and that product is known as propionyl-CoA. Then in order to make propionyl-CoA as something that the body can actually handle with existing cycles, the cell will carboxylate propionyl-CoA with the use of ATP and biotin to form methylmalonyl-CoA. Well, the methylmalonyl-CoA will then be isomerized to succinyl-CoA. And then succinyl-CoA, by now you will click right away, will enter the TCA cycle and be further converted to oxaloacetate and further degraded. All right, so that basically covers the reactions of beta oxidation for saturated, mono, poly, and unsaturated, and for odd number fatty acids. Now let's look a little bit into the regulation. All right, so let's go over these questions. It's the beauty of the in-class presentation is that I will not show you all the questions at once, but we'll still make good sense of it. All right, so what determines if a fatty acids will be used for synthesis or be degraded? Remember, when the fatty acid is in the cytosol, it can be used for biosynthetic purposes for membranes. And the membranes are constantly regenerated, so a lot of fat goes for membrane regeneration or membrane generation in general. Now, what would determine if that fat will go for synthetic purposes or for degradation? The answer is energy needs. Do we need energy or not? And if the fatty acid is bound to the carnitine, which is the shuttle that moves fatty acids from the cytosol to the mitochondria, it's shown right here. Basically, the fatty acids is bound to carnitine, forming fatty acylcarnitine, and then that moves the fatty acids inside the mitochondria, and that's where beta oxidation occurs. And on and on and on. Acetyl-CoA generation, and then the citric acid cycle. So, energy needs and whether or not the fatty acid will be bound to carnitine. Okay, so you can see here on this diagram that excuse me, that malonyl-CoA regulates the carnitine shuttle. So let's look into that. What do you think the effect of malonyl-CoA would be on the carnitine shuttle? So obviously you figure in the textbook will show you immediately what that is, but I've hidden it here so that we can make sense of it. And by reasoning it, by understanding it, you don't have to memorize what it does, but you'll be able to derive it. Okay, so malonyl-CoA would regulate the carnitine shuttle. So what is malonyl-CoA? Well, malonyl-CoA is the first product of fatty acid synthesis. The first product of fatty acid synthesis. Now, what makes malonyl-CoA? It's an enzyme known as acyl-CoA carboxylase. This enzyme gets activated by a phosphatase that dephosphorylates the enzyme and activates it to convert acetyl-CoA into malonyl-CoA. the phosphatase is activated by the insulin receptor signaling pathway. When is the insulin receptor or insulin hormone released? When we have high blood glucose level, when we have consumed food. So you consume glucose, we stimulate insulin, insulin stimulates glycolysis, glucose will be degraded to to pyruvate, and then from pyruvate to acetyl-CoA. At the same time, insulin stimulates, dephosphorylates ACC enzyme that will start converting acetyl-CoA to malonyl-CoA, forming fatty acids. That means that we have excess of acetyl-CoA. There's plenty of it, so we can store some of it as fatty acids. Now, what do you think malonyl-CoA would do to the carnitine shuttle? Stimulate it or inhibit it? Do you want to degrade fatty acids to generate more acetyl-CoA, or you don't? Well, if you have plenty of acetyl-CoA, so much that you're even storing that acetyl-CoA as fatty acids, then it will not make any sense to break down fatty acids so that you will generate acetyl-CoA for energy. So the effect of that of acetyl-CoA would be to inhibit the carnitine shuttle. All right. Next question. Why do you think beta oxidation is stimulated when blood glucose level is low? Well, I mean, we kind of already talked about it. You need energy. You don't have sufficient glucose, so you want to shift, the body wants to shift its energy production from fat, from deposits. It'll stimulate, glycogen breakdown, gluconeogenesis, and it will start using fat for energy. So that will be the purpose of it. It wants to spare in glucose so that the glucose will be primarily sent to the brain. All right, so that's the purpose. Why we want to stimulate beta oxidation when blood glucose level is low. I mean, right here also, glucagon, on the other hand, will, through the protein kinase A, will phosphorylate the ACC enzyme. The enzyme will simply not be available to convert acetyl-CoA to malonyl-CoA. Loss of malonyl-CoA would lose the inhibition, or the inhibition will be lost for the carnitine shuttle. Now the carnitine shuttle freely can start moving fatty acids into the mitochondria that will be degraded to acetyl-CoA and for energy. So it makes sense. You can see the balance. All right, here's another question. When blood glucose level is low, meaning that there is no malonyl-CoA to substitute, to inhibit the carnitine shuttle, glucagon is stimulated, glycogen degradation is stimulated, fatty acid degradation is stimulated. How would beta oxidation be affected if you drink 1 can of regular soda pop? Now, if you think about it, why do I say regular and not diet? Well, that means, if regular soda pop has about, if I'm not mistaken, about 25 grams of sugar, I mean, that's a lot of carbohydrates. Now, if you just drink 1 can, that's fine, but, if you get a, supersize soda pop, or even if it's orange juice or whatever, any drink that is with high glucose in it and good quantities of it, that glucose immediately will spike the blood glucose levels, because this is a simple carbohydrate. It's a disaccharide, if you remember. Sucrose is a disaccharide. It just takes 1 degradation and it becomes glucose and fructose, which are basically feed into the same pathway. That spike of glucose in the blood would immediately activate insulin, will shut down glucagon. Glucagon will basically, if I can use a previous slide here, glucagon will stop the mobilization of fat from the fat deposits that normally would export the fats for beta oxidation. When you increase the blood glucose level, that's going to stop. At the same time, you're stimulating insulin that stimulates glycolysis. also ACC conversion that's going to convert acetyl-CoA to malonyl-CoA if you have sufficient amount of acetyl-CoA. I mean, because a lot of it is going to go into the TCA cycle, especially when your blood glucose level is low. But still, I'm sure some of it will be generated if you take enough glucose. Some of it probably will generate malonyl-CoA. But just simply because we're shutting down glucagon and we're stimulating insulin, this process will be inhibited. So you would stop the use of inhibit the use of fatty acids for energy. You will transition now to use glucose now for its energy. Now, what's the problem though with all of this? Someone would say, oh, good, you got the glucose, now you don't need to use any more fatty acids. Well, the problem is, to use a quote from a favorite show of mine, you haven't eaten yet. So anyway, you actually, or a person that would do that, if you're hungry, if you have low blood glucose and you just drink a soda pop, a classic soda pop, or some form of a sugary drink, you are not really consuming any food. You're basically taking concentrated amounts of glucose that will immediately spike your glucose, spike insulin, and then the result of that would be insulin will quickly decrease the glucose in the blood. That's going to make you actually hungry after a little bit. So it's almost like a vicious circle. And you take a lot of calories in the form of glucose. You're shutting down the use of fats from your own body, and that's how you end up increasing actually your fat deposits rather than shrinking your fat deposits. So when sometimes people say, you shouldn't eat fat so that you don't get fat, I was actually, I don't know how many of you, probably none of you remember the 90s, the late 80s and the 90s, but there was this big deal about fat, fat-free this, fat-free that, everything was fat-free. I even remember seeing a fat-free butter, a corn butter. I was wondering, I was like, then what is it? If it's butter and it's fat-free, then what is it? And there was this big deal, don't eat fat, because fat's going to make you fat. And then at the same time, people focus on sugar, and they keep drinking sugar drinks and massive amounts of other sweetener or sweet products. And of course you will not decrease in size. Of course your fat deposits will continue to increase because of this. We haven't talked exactly about fatty acid synthesis, but you can get a glimpse of it right here. Acetyl-CoA is a natural product of glucose degradation. One of the products of acetyl-CoA is malonyl-CoA, that is the first compound that enters fatty acid synthesis. You don't need to eat fat to generate fat in the body. All you need to do is eat carbohydrates, and you will increase in size. You don't have to have a molecule of fat, and you will still get fat because of this pathway. Anyway, I think I sidetracked myself, but as I've told you, the relationship between carbs and fat is one of my favorite topics to learn about and to teach, and so you can tell that I can easily be sidetracked into all of that. But anyway, to get back to this, when you have low blood glucose level, if you just take a drink that has concentrated sugar, all of this will be stopped, and you will shift the body will immediately shift its energy usage from glucose and not from fat. Okay, now we're going to go into ketone bodies now. I want to ask you first, when are ketone bodies generated? And the answer to that is ketone bodies are generated when the body is starving. And not right away even, but after a while, you will start, at some point I will show you in one of my future lectures, I think it's actually next chapter, that you start seeing a measurable or significant increase after about 48 hours of starvation. That's when they're generated. So ketone bodies are generated when the body is starving, there is no glucose, and this is in a way a substitute for the brain and other tissues when we don't have glucose but we're degrading fat. And so the fat gets converted to acetyl-CoA, as you already seen, and that acetyl-CoA, one way to use it for the body is to convert it into ketone bodies. We have 3 ketone bodies: acetone, acetoacetate, and beta-hydroxybutyrate. They're very much connected. If you see the difference between acetone and acetoacetate is one carboxyl group added to one of the methyl groups of acetone. And the difference between beta-hydroxybutyrate and acetoacetate is instead of a ketone group here or a carbonyl group, we have a hydroxyl group bound to it. So these are very close structurally compounds. All right. Again, the point of ketone bodies is to substitute for glucose to feed the brain and other tissues. So how do we generate ketone bodies? It's actually very simple. You take 2 acetyl-CoAs. With the help of thiolase, we make acetyl-acetyl-CoA. Acetyl-acetyl-CoA will accept another molecule of acetyl-CoA, and with the HMG-CoA synthase will form HMG-CoA. HMG stands for beta-hydroxy, beta-methyl glutaryl-CoA. You can see why I prefer HMG instead of beta-hydroxy, beta-methyl glutaryl-CoA. It's a mouthful. So HMG-CoA, then an acetyl-CoA is removed and we end up with acetoacetate. So the difference between acetyl-acetyl-CoA and acetoacetate is just that CoA group. So what happened so far? We basically condensed 3 molecules of CoA and we removed 3 molecules of acetyl-CoA, and then we removed 1 acetyl-CoA to end up with acetoacetate. And of course, in the process, we remove 2 molecules of CoA. Okay, acetoacetate is the first or the main ketone body. If we decarboxylate it, if we remove that carboxyl group, we end up with acetone. And if we reduce it, we get beta-hydroxybutyrate. You just see, instead of a ketone group here, a carbonyl group, we end up with a hydroxyl group. So beta-hydroxybutyrate. Now, beta-hydroxybutyrate and the acetoacetate, but specifically, well, primarily beta-hydroxybutyrate, then all of them actually, all 3 of them they're released in the blood, they can pass through membranes, and that's the beautiful thing about the ketone bodies, is that when they can pass through the membranes, and then when they reach their target cells, then the beta-hydroxybutyrate gets oxidized to acetoacetate. Basically, we're going backwards now, but in a different cell. This happens in the liver. This happens in the target cells, like brain, kidney, muscles, anything. Then we get to acetoacetate, then succinyl-CoA, beta-keto-acetyl-CoA transferase. We convert the acetoacetate to acetyl-acetyl-CoA, and then with the help of thiolase, we generate acetyl-CoA. So what happened here? I missed to tell you, but I'm going to tell you right now, this process of generating ketone bodies ketone bodies happens only in the liver. This happens only in the liver. No other tissue will generate ketone bodies. That's why the liver is so essential. And the liver is the only tissue that cannot use ketone bodies. So the liver generates ketone bodies. They're released for every other tissue to use. So that's why I'm repeating this again, because the generation happens in the liver, then beta-hydroxybutyrate and acetoacetate are released. They go back to acetylacetyl-CoA, and then from acetylacetyl-CoA to acetyl-CoA. Now that acetyl-CoA in those tissues will be used in the TCA cycle to generate energy. That's the beauty of this process. And to add to it, I want to ask you, did we use any ATP? Someone might say, what difference does it make whether we use it or not? Well, think about it again. When do we make ketone bodies? During starvation. The body desperately needs ATP. The beautiful thing, again, of this process is that no ATP was used. The cell has preserved its ATP, and the process takes acetyl-CoA from the liver, repackages it, or packages in the form of ketone bodies, sends it out, and that ketone bodies data is degraded to acetyl-CoA, but now in the target tissues, those that actually are starving and need energy. So that shows the importance of the liver and the beautiful process of ketone body generation. Now, the ketone body generation, even though they're great and they supply the tissues that are in need of energy with necessary energy, ketone bodies are not the most friendliest molecules in the body. I don't know whether any of you has used acetone, but you know how reactive acetone is. These are not friendly molecules in terms of all the other compounds around them. Of course, they're at a very low concentration, but still, these are toxic molecules. As a matter of fact, they're also acidic. As a matter of fact, if you have too much ketone bodies in the blood, you get into a condition known as ketoacidosis that can lead to coma and death. So even though this is a beautiful process, this is a process that is reserved for the basically a time where there's nothing else available. No carbohydrates or proteins are available, and the only source of food is fat. And as a result, the body or the cells are now adapting to use products of fat degradation for energy, since there is no carbohydrates to be used for energy. And again, that's the basis of a lot of the fat and protein-based diets. The so-called the keto diet, for example, or in the past, whether you've heard it or not, I'm not sure, but the so-called Atkins diet. And I remember people were so surprised when when he came up and says you can eat bacon and fat and butter and whatever you want and you'll lose weight. And people were like, what? How is that even possible? Well, because of that, you starve the body from glucose. The body will shift into degrading fats, especially its own fats, forming ketone bodies that now will feed the brain and all the other tissues. But you have to be careful. Not that this would, in normal conditions, this probably will not happen. If you're a healthy individual, you will not get into a ketoacidosis because the body can regulate the concentration of the ketone bodies. But still, you get into this state where ketone bodies are produced and now they're used for energy. And that's how you lose weight. That's how you will eat butter and fat, all other fats, and meat, as long as you don't have carbohydrates, and you will lose weight. That's the premise of these diets. Anyway, I could talk about this for a while, but I think I should move on. All right, so why ketone bodies are synthesized under low blood glucose levels? It's a great question. I think I pretty much answered it, but I really love this figure because it shows in a greater detail why this is actually happening. All right, so when we have low blood glucose level in the liver, we're talking about the liver right now, that stimulates gluconeogenesis. Why? Because the liver is the only tissue that can produce glucose that will be exported out for the brain and other tissues to use. Yes, but what is the source of gluconeogenesis? Someone might say, well, pyruvate. Yeah, but how will you generate pyruvate? Pyruvate is only generated from glucose. You don't have glucose. So how will you generate it? Well, there is a precursor that can actually circumvent pyruvate, and that's oxaloacetate. If you remember, if you look at the gluconeogenesis pathway, oxaloacetate is part of the gluconeogenesis pathway. Oxaloacetate is now used by the liver to generate glucose and exported out for the brain. But oxaloacetate is also used in the citric acid cycle. What would happen with the TCA cycle if you start pulling away oxaloacetate, which, by the way, if you remember, has very low concentration because the malodehydrogenase reaction is an unfavorable reaction that is followed by a very favorable reaction, which is citrate synthase. So not only now we have a situation where malodehydrogenase doesn't want to produce oxaloacetate, and then whatever oxaloacetate is produced enters the TCA cycle because we need energy, but also oxaloacetate is pulled away for gluconeogenesis. Now, what do you think, how would that affect the citric acid cycle if you pull away oxaloacetate? It would inhibit it. Maybe not stop it, but it will inhibit it, in the liver at least. At the same time, the fatty acid degradation is stimulated. Well, what's the product of fatty acid? Acetyl-CoA, available to be burned in the citric acid cycle, which we just said was inhibited. So now we're breaking down fatty acids. The cell is dependent on the electrons released from beta oxidation. But in order for beta oxidation to continue, you need a continuous supply of CoA to generate acetyl-CoA. Yeah, but acetyl-CoA is not used now, or not as well used in the citric acid cycle. So eventually the CoA will be depleted, which will stop fatty acid oxidation, beta oxidation, which will stop the energy for the liver. It's a problem. So what do we do with that? We have to somehow release CoA to go back to beta oxidation. But then what are we going to do with the acetates? Well, we'll fuse them together and form ketone bodies that don't have CoA. Then the CoA comes back. It gets reinvested in beta oxidation. More acetyl-CoA is generated, that even though part of it will enter the citric acid cycle, there's way too much because the citric acid cycle is inhibited. So that excess of acetyl-CoA will be used to generate ketone bodies, release CoA. The ketone bodies now can cross the membrane. The hepatocytes enter the blood and then be used from all other tissues except the liver, brain, skeletal muscle, kidneys, to do their function. So that is how beta oxidation, gluconeogenesis, ketone bodies, ketone body production, they're all connected so that we will continuously provide energy for the brain and other tissues when we don't have sufficient amount of glucose in the blood. Well, here's the follow-up question then. Why are ketone bodies then synthesized during diabetes? What is the main symptom of diabetes? High blood glucose level. Wait a minute now. We just said that ketone bodies are synthesized when blood glucose level is low. So how come we're going to synthesize ketone bodies when the blood glucose level is high? Well, even though I'm trying to confuse you with these questions, you probably already realized that what's the problem with diabetes? Why is there actually high blood glucose level during diabetes? Because there is no insulin. The insulin cannot help the glucose enter the cell. So for all intents and purposes, the cells are bathing in glucose, basically. There's tons of it, but they cannot use it because the glucose cannot enter the cell to be degraded and be used for energy. So for that reason, the body is actually, the liver now is starting to pull away oxaloacetate to generate glucose. And I don't know to what extent gluconeogenesis is activated in this process, because we still have high blood glucose level, but that glucose cannot enter the brain or other cells, or not enter it at least very effectively. And so I would imagine that this process is still activated to a point, which makes this even worse, because all that newly generated glucose probably would even make things worse in terms of increasing the blood glucose levels. Regardless to what extent gluconeogenesis is activated, though, the body still needs energy, and fatty acid degradation will be stimulated. And there will be way more acetyl-CoA than the citric acid cycle in the liver at least can handle. That acetyl-CoA now will be packaged into ketone bodies, and they will be released. So even though we have high blood glucose level, we also have high ketone bodies level in the blood, because that is the only way. They don't require transports. They can cross the membrane, the cell membrane, and become available for the cell to use now the acetyl-CoA generated from fatty acid degradation, and the tissue can use that now, acetyl-CoA, for their own energy needs in the absence or when glucose level is very low. So hopefully this was interesting to you. Hopefully you actually understand how this works. This is perhaps one of the most informative diagram that I have seen, at least in textbook, that kind of helps me see fatty acid degradation, glycolysis, gluconeogenesis, ketone body production, all of that connected together, and then how is it used by the body to facilitate its own energy needs. And with that, I will end my presentation. Like I said, I hope this was informative. This was a little bit of a longer lecture. It's pushing 1 hour now. But I enjoy talking about this subject, so I'm sorry that I took more time than I normally would for a regular class. But I hope this was helpful. Thank you for your time.